Very good evening, everyone. Am I audible? And is the PPT visible to you? All, please confirm. Excuse me. Okay, Umara. So, today we'll be talking about early development in model organisms. We'll be seeing sea urchin only. Okay. So, let's begin with the first question. This is regarding sea urchin. During normal development of sea urchin, beta-catenin accumulates predominantly in the micromeres. So for these, you need to know all types of cells which are there. Okay, what all cells are present in the micromeres? They have already mentioned that beta-catenin predominantly in the micromeres, and those are fated to become. That means whichever cells in the sea urchin, that is in the micromeres, wherever it is beta-catenin is present, those micromeres, they are fated to become endoderm and mesoderm. So the question is, if GSK is blocked in the developing embryo, if GSK3 is blocked in the developing embryo, then what out of these will be the outcome? We have been discussing about sea urchin early development, access specification, the classes in our Biotechnica classes under developmental biology. We have been studying this. So now is the time that you apply your knowledge and answer the following questions. Those who are not part of Biotechnica classes, if you are doing self-preparation, then you can also try this. Anyways, I will be providing an explanation for the same, but before that, you all must give it a try. So you can just let me know if you're solving. So I'll be waiting for you to answer. Okay, Achal, let us see whether your answer is right or wrong. We'll wait for others also to give the answer. So anybody else wants to attempt? Please let me know if you are trying to attempt this question. In that case, I'll wait for some more time and explain you the answer. Excuse me. Okay. So yeah, Achal, your answer is right. Very good. Correct option is answer 3. Let me explain how. Okay, Reshma, anyways, I have mentioned the answer. So beta-catenin will accumulate. Yeah, Aishwarya, I'm explaining here. It will accumulate in the nuclei of all blastula cells, leading to animal cells, getting specified as endoderm, mesoderm. So for this, you need to first understand about the beta-catenin pathway. Okay, beta-catenin and GSK pathway. So as I mentioned that in this particular signaling of beta-catenin and GSK3, So you have a receptor which is frizzled receptor, and in case the WNT signaling is there, they activate another molecule called as DSH. If this DSH is activated, they will inhibit a complex formation, that is GSK, APC, and other protein formation. If these are inhibited, then there is a molecule which is beta-catenin. It's a transcription factor. So it is free. Initially, they were all bonded together. But because of DSH, this complex formation is blocked. As a result, beta-catenin is free to enter in the nucleus. And there it is free to transcribe the gene. So beta-catenin binds with some regulatory regions of the gene sequence and transcription is activated. This is how it works. So what does the question say? That if GSK is blocked, that was the question. Now already WNT signaling, if it is there, through this signaling GSK anyways will be blocked. But in your embryo, if the question is saying GSK is blocked, that means signal or no signal, it will act as if there is a beta-catenin in the cell. That means wherever cell GSK will be blocked, that cell will have beta-catenin. Okay, this is the logic. Now what is said? Beta-catenin will accumulate in nuclei of all blastula cells. Beforehand, the series of cells that you have in sea urchin, so you have the tier 2 micromeres. Okay, micromeres are actually cells of the blastula. We know that when a zygote forms, it gives rise to blastula. So in sea urchin blastula, you have tier 2 micromeres, which are also called as small micromeres. Then you have tier 1 micromeres, tier 1, which are the large micromeres. Then you have a layer of wedge 2 cells. Then you have a layer of wedge 1 cells. Then you have AN2 cells, and then you have AN1 cells. So generally, this beta-catenin, which will activate the gene transcription and make the cell fate undergo endoderm and mesoderm. Whichever cell will have beta-cat, those will be forming endoderm, mesoderm. So generally, those are present in these micromeres, especially the large micromeres, and to some extent, the wedge 2 cells. Okay, but how beta-catenin was present in these two layers of cells? It was present because of the WNT signaling, which was there only in these cells. Okay. But now the question is saying the entire embryo GSK is blocked. Here in these cells, GSK was blocked, because of which beta-catenin was free. But that was because of the signaling. Other cells, GSK was active. That's why here beta-catenin was not free. Okay? But now, since in the entire embryo GSK is blocked, even in these cells, beta-catenin will be free. So all the cells of the embryo, whether signaling is there or it is not there, beta-catenin will be present. And if beta-catenin present in all the cells will be fated to become endoderm and mesoderm. So in such kind of cells, you will not find ectoderm. Everyone got it? Did you understand the explanation? So as you know, in our Brahmastra series, we are discussing CSIR questions, right? So as you can see, this topic is very, very important. So many questions are coming. Beta-catenin accumulation in the nuclei of large micromere will be inhibited. Why? If GSK is blocked. So anyways, beta-catenin accumulation will be there, whether signal or not signal. So why will it be inhibited? Not correct. Beta-catenin accumulate in nuclei of all blastula cells, leading to an ectodermal wall. Till here it is correct. It will accumulate in nuclei of all blastula cells. But whichever cell it is present, it will lead to the formation of endoderm and mesoderm. Why will it form ectoderm? No reason. Beta-catenin in accumulated nuclei of all blastula cells, because of which all the animal cells will get specified as endoderm and mesoderm. Again, fourth is saying it will accumulate in nuclei of large micromere will be inhibited, not correct. Okay? Yeah. Okay, Reshma and Surinder. Yeah. Achal, you gave the correct answer. Very good. So see here, this is what I told you, that if GSK is blocked, in some other question you may get this. So the agent which blocks beta-catenin is which blocks GSK3 is lithium chloride. So as we discussed in the classes, if you have the lithium chloride treatment, then they block GSK. And Disheveled blocks GSK, because of which beta-catenin gets free. So anyways, if GSK is blocked, beta-catenin will be available in all cells. And that will lead to the specification of endoderm and mesoderm. Okay. If you have a regular embryo, a normal embryo, you will see that beta-catenin, the orange colors are representing that. They have accumulated in the large micromeres, small micromeres, and somewhat in the Wedge2 layer. So only these cells, they have made endoderm and mesoderm. In the cells where beta-catenin is not there, that is the mesomeres and Wedge1 cells, those have become ectoderm. And these have become mesoderm and endoderm. So overall, a wild type embryo has formed. This is a Plutus larva. Okay. So I'd just like to remind you about the Raftar batch. As you know, we have opened the admissions and it will be starting from 11 September 23. So what are you getting in the Raftar batch? So it's speed revision batch. Okay. So in case you have been preparing but you want a revision, fast forward summary revision, this is the best package that you can get. Because along with this Raftar batch, yes, classes will be there where you are getting the quick revisions taking place for December 2023 exam. Along with that, you will also get the portal access. If any student wants to enroll, you don't want to enroll for regular class, just want revision, then this is the best package for you, provided you already have the foundation concepts. If you're just a beginner, then I'll suggest you join the regular batch. Okay. So you will get all the PowerPoint presentations that the teachers are using. You will get daily practice sheets, questions, faculty support 24/7, Animatica Zone, which contains 100 plus animations. You will be able to see them. It's always better to study with visuals. You will also get the test series AIMNet, which is unit wise. Okay, so you will get unit wise. If you want to solve only unit 1 questions, you will get an AIMNet test only for unit 1 questions. Along with that, you will also get, this is the best part, you will get access to Biotechnica's revision tools, which I keep telling. That is concept wheel, concept table, conceptica flowchart, concept cloud. So all those revision tools, you will get access. Okay. But we have limited space, as you know, seating limited, seats available. So you must contact fast. This is our toll-free number, and this is our office number in case you want to contact in office hours. Let's move to question 2. Again, we are talking of sea urchin, similar concept. Since I already explained to you, this you should be able to answer very quickly and clearly. So there are some few statements regarding DSH. Just now we were discussing disheveled and beta-catenin in development of sea urchin. A and B says that DSH is localized in vegetal cortex of oocyte, whereas B says DSH is localized in cytosol of oocyte during oogenesis. C says it predominantly accumulates in micromeres and somewhat in wedge to tear. D says if you treat with lithium chloride, it does not allow accumulation of beta-cat. And when E says beta-cat is prevented from entering nucleus, the embryo develops as a ciliated ectodermal ball. So you already know the concept. All you have to do is now apply it and tell me the correct answer. At least let me know that you are solving so that I'll wait for your answer. Else, I will just simply explain. Okay, Achal, let's see. Okay, what about A and B, Surinder? Between A and B? Okay, Saujanya. So see everyone. Anyways, okay. Aishwarya also says option 3. Surinder says option 1. Reshma is confused between A and C. A and C are very different kind of statements. If you are confused between A and B, then that makes sense. Because C, anyway, C is correct. What did I show you just now in the previous diagram? Didn't I tell you that beta-catenin accumulates in the large micromeres, means in the micromeres, and to some extent in Wedge2 cell. I already told you, right? Beta-catenin predominantly in the micromeres and lesser in the Wedge2 tear cells. So anyway, C is correct. Right? So C is correct. Then next we have to see between A and B and D and E. So DSH, whether it is localized in cytosol or in vegetal cortex. Now remember I told you the beta-catenin pathway, that wherever DSH will be activated, there GSK will be blocked, and that's why beta-cat will be free. So indirectly, wherever DSH is there, beta-catenin will be free. So if beta-catenin is appearing in micromeres and Wedge2 cells, where are these located? The micromeres and Wedge2 layer of cells, where are they located in the entire embryo? That's right, Achal. They are located in the vegetal pole. Yes, correct. Achal, Surinder, yes. Manishri and Surinder, very good. Your answer is right. Okay. So, yes, they are in the vegetal cortex. So, DSH will also come in the vegetal cortex only. It will not come in oocyte. It will not come in entire cytosol. If entire cytosol has DSH, then all the cells would have DSH activated. But that's not the case, okay? They are localized only in the vegetal cortex of the oocyte before fertilization and in the region of the 16th cell, which is about to become the micromeres. Okay? That's how it somewhat appears in the Wedge2 layer also. Okay? So A is correct, C is correct. Now we have to see between D and E. So this also I mentioned. If lithium chloride is there, so tell me, what is the purpose, what is the property of lithium chloride? What does it do? What does lithium chloride do? Just now we had discussed in the previous question. That's right, Nachal. Very good. Yes, Surinder also right. Very good. It blocks GSK. So if lithium chloride is present in embryo, yeah, Vanishri also right. Very good, all of you. Treatment of embryo, yes, Aishwarya also right. Very good. So it's not allow expression of beta-catenin. It will make it free. Okay. Beta-catenin is already present. Beta-catenin will be free to enter nucleus and it will allow expression of other genes, not beta-catenin expression. Okay. Yeah, that's right. Reshma, it blocks GSK, not GSM. Okay, GSK. It's a kinase enzyme. which in turn leads to beta-catenin being free. Very good one, Ishri. So GSK, if it is blocking, it will lead to beta-catenin being free. But what is it writing? Does not allow the accumulation of beta-cat. It will be opposite. It will allow the accumulation of beta-cat. That's why this is wrong. Okay, it's written it does not allow accumulation of beta-cat in nuclei of all blastula cell. No, it allows it. And the animal cell becomes specified as endoderm and mesoderm. This is correct. Only this does not word is not correct. So D is not correct means E is correct. So yeah. Oh, I think by mistake I have marked it. Yes, correct answer is option 3 only. Okay. Right. So let me see who gave the right answer. Yeah, Achal, your answer is right. Yeah, Aishwarya also gave the right answer. Very good. Okay. Clear, Vanishri, Surinder. So it will be vegetal cortex. Okay. And it is present before fertilization only. Okay. So why E is wrong? E is correct when beta-catenin is prevented from entering nucleus. So even though in the vegetal cell, veg2 and micromere, even though the beta-cat is present, but if you don't allow it to enter nucleus, then what's the point? So beta-cat should be able to enter nucleus, then only gene transcription will be there, right? So if you, then only endoderm, mesoderm will form. So if you don't allow beta-cat to enter, then these cells will form ectoderm only. They can't form endoderm, mesoderm. Okay. So this diagram which I showed you, this one, inhibition of beta-cat transportation into nuclei. So in that case, beta-catenin cannot enter nucleus. It will be limited only in cytoplasm of the small micromeres, and Veg2 cell, it will be limited only in the cytoplasm. So as a result, what you will get? You will get a ciliated ectodermal ball like this. Okay. So this is your beta-catenin pathway. As I mentioned, you have the WNT, which is the signal name. It binds with the frizzled receptor. Upon activation of frizzled, they will activate another protein called Disheveled. Disheveled blocks GSK APC axine kinase. So if that complex was forming, then beta-catenin would have been degraded. But because this complex is inhibited, beta-catenin is free. It is free to enter the nucleus. It binds with other transcription factors and it will associate with the regulatory region of the gene. So beta-catenin specifies the vegetal cells. Micromeres undergo autonomous specification. As we know, the large micromeres, they are autonomously specified by this beta-catenin to become skeletogenic mesenchyme. And these micromeres produce the initial signals that specify other tier of cells. So micromeres will give signal to the Veg2 cells. Veg2 cells will give signal to the Veg1 cell. Like that it will happen. Molecule responsible for specifying micromere is beta-catenin. The accumulated nuclei of those cells fated to become endoderm, mesoderm, and this accumulation is autonomous, which is responsible for specifying vegetal half of embryo. And during oogenesis, DSH becomes located in vegetal cortex of egg. This is the explanation, what I just told you. Okay. This is regarding the double negative circuit. Double negative circuit of gene regulatory network of sea urchin only. That is, two cells they have given, micromeres, and rest of the cells of blastula. So just now I told you that micromeres are autonomously specified. We have talked about this question. We have talked about this particular topic today in class. So just have a look at this diagram. We have also studied about PMAR, HESC, right? So here they have a column 1 and column 2. Column 1 is what they are injecting. So in that you have mRNA of PMAR1, mRNA of HESC, antisense of PMAR1, and antisense of HESC. And development outcomes is, one is all cells start ingressing into blastocele, and second is skeleton mesenchyme will not be formed. So you have to match with A, B, C, D with 1 and 2 and give the correct answer. So as you can see, there are some set topics from which questions come. Okay. But merely studying those topics, first of all, if you do self-study, then it will take forever for you to understand these concepts. They can be really confusing for beginner students. Okay. And even if you are registered for biotechnica classes, so as you all will agree that you understood everything in the class. All this we have learned today, right? Definitely you people have understood also. But when it comes to solving questions, you have to not only remember the concepts, but also you have to apply them in different ways. So the more you solve the questions, the more you attend such sessions like this, okay, the more you will get better in part C type of questions in CSIR exam. Okay, we'll see, Surinder. Okay, Achal also says 3. Is anyone else trying or should I explain? Okay, fine, Anupam. So yes, all of your answers are right. Very good. Correct answer is in fact option 3. So congratulations to you all, since you were able to solve the question very efficiently. So, Surinder Achal Anupam, did you see this question before? Or you have solved it based on today's class explanations? Based on class, that's good to know. That's good to know, Achal. So keep paying attention like this. It means that you are on the right track. Okay? Because not many people are able to solve in the first attempt after attending the class. Okay? So keep it up. Column 1 lists the experiments carried with mRNA or antisense RNA. Now, mRNA means what? That it is present. Antisense means what? It is not present. Okay? Because antisense is blocking the actual mRNA. So that's why the mRNA is not active. Okay, so this is the concept you need to know from unit 3. And from here, sea urchin, you need to know the concept that in micromeer, anyways, we know beta-catenin is there. So they will activate PMAR1. Okay, so micromeres are autonomously specified. It's because of beta-catenin. And micromere, they are supposed to give rise to skeletogenic mesenchymal cells, the ones which will give rise to skeletons, skeletal rods in sea urchin. So if PMAR is there, then PMAR inhibits HESC. And if HESC is present, so HESC generally will inhibit, if you see here, in other cells where beta-catenin is not there. That's why PMAR also is not there. So PMAR cannot inhibit HESC, so HESC will be present. In those other cells, HESC blocks the transcription factors needed to form skeletogenic mesenchymal cells. Okay. If HESC is there, then they inhibit the formation of, that is the transcription factors that is forming skeleton. Which means rest of the cells of blastula will not make skeleton. Okay, because HESC is blocking them. But micromere, in the micromere, because of beta-catenin, PMAR is there. Because PMAR is inhibiting HESC, HESC cannot inhibit this. That's why these transcription will be happening. And because of this transcription, skeletal will form. Skeleton will form. So simple, they have already given the logic. Based on that, you have to just solve the question. So if PMAR is there, which means HESC is inhibited. HESC is inhibited means all cells will start ingressing into blastocele to form the skeletogenic mesenchyme cell. So we know mesenchyme means the cells which come out of epithelium. They will come out as individual cells, and those cells only form the mesenchymal cells. These are called as loose cells. This is epithelial layer, and from here they come out as loose cells. These loose cells later join together and form the skeletal rods in Plutus larva of sea urchin. So if PMAR is there, mRNA of PMAR, all cells, irrespective of whichever, because if PMAR is there in all the cells, HESC will be blocked. In that case, all cells will form skeleton. Because HESC is not there means In all cells, the transcription factor for skeleton formation will be activated. All cells will start ingressing into the blastocyst. A will be 1. If you put mRNA of HESC, okay, so in that case, wherever you have put mRNA of HESC, so that HESC will go and block the transcription factor and skeleton mesenchyme will not form. So B will be 2. If you put antisense of PMAR, it means PMAR is not there. If PMAR is not there, it means nobody can block HESC. So HESC will block skeleton formation. So C will be 2. Skeleton mesenchyme will not be formed. If you put antisense of HESC, antisense of HESC means HESC not there, which means nobody can block transcription factor, skeleton will be formed. So all cells will start ingressing into blastocyst. Everyone got this? Those who could not answer, now did you get this part? Please respond. So fun fact is that sea urchin topic is not mentioned anywhere in your developmental biology syllabus of CSIR. Nowhere it is mentioned. But still you get, as you can see, all these are CSIR questions only. Okay. So this inside things Biotechnica tell you, because we have experience what all questions come and not. We know from what area it is coming. And we teach in class those topics. And we also discuss the questions. So this is summary. In micromeres, activated PMAR represses HES repressor and micromere specification signal genes are active. Whereas in Veg2 cells, PMAR is not activated. Because of that, HESC shuts down skeletogenic genes. And those cells, they respond to the notch signal. Containing notch, they respond to delta signal to form the skeletal mesenchyme. This we'll discuss in next class. I'd like to remind you about the Raftar batch. So you must hurry up because the last date is to register will be in September only, starting from 11 September. So don't miss from the first class itself. Next question. In response to a WNT signal, beta-catenin enters the nucleus and binds to the LEF1 TCF protein by displacing which of the following proteins? Yeah, let's see, Anupam. So as I mentioned, beta-catenin is transcription factor which is activated because of WNT signaling. And if beta-catenin is free, it enters the nucleus and it will bind with the other transcription factors, that is LEF1, TCF1. But while binding with these other factors, it will replace some other protein also. So which out of these it will replace? And instead, the LEF1 TCF, instead of binding with that protein, now it will bind with beta-catenin. Okay, Aishwarya. So yes, very good. The correct answer is Groucho. So this is actually part of the signaling chapter. Anyways, very soon in our classes, I will be conducting a separate additional class for signaling pathways in developmental biology. Because Unit 4 and Unit 5 goes together. You get some related questions from these two units. Okay. So this is the explanation. So as you can see, in absence of ligand, we have the frizzled receptor and DSH not bonded with each other, because of which all of this form a complex, and the beta-catenin is phosphorylated, excuse me, because of the GSK, and because of phosphorylation, beta-catenin can be ubiquitinated and degraded. This is an absence of ligand. The transcription factors, so we have TCF, CBP. So TCF is occupied by Groucho, because of which the transcription cannot proceed. Target genes are repressed. Okay. So they will repress it in association with Groucho and CBP also, but mainly Groucho. Groucho acts as inhibitor. Okay. But if you have the ligand, then these all become free. They can't form the complex. As a result, beta-catenin cannot be degraded. So it becomes available in the nucleus. So there it will replace the Groucho and itself will bind with TCF. Now that TCF is associated with beta-catenin, it will allow the target genes to be activated. So that's why correct answer is Groucho. So we have one more question. Now I expect everyone to answer this correctly. So we have already seen enough about sea urchin. Another question was there, which I'll discuss it in next class because it will take time to explain that particular question. Following statements with respect to development in sea urchin. Self-fates are determined both by autonomous and conditional modes. Large micromeres are conditionally specified. Large micromeres produce paracrine and juxtacrine fractals that specify fate of neighbors. So as you can see, some signaling concepts are present here as well. Beta-catenin is not required for specification of micromeres. You have to select the combination with all correct answers. Surinder has given me an answer. I want answers from rest everyone as well. Okay, Vanishri. Okay, Reshma. Okay, Anupam. Fine. Yes. Many of you are there. Others also. Tell me what will be the answer. So should I explain, or you are still, any one of you is still trying? Please let me know. If not, then I'll explain this. Okay, Anupam. So see, first of all, based on what today's concept we learned. Today we learned a lot of concepts, right? So at least you will understand what beta-catenin is not required for specification of micromer. Is this true or false? You all should have given correct answer. And you will give correct answer now also. I will just lead you to the answer. Tell me. So you all also solve like this only, how I am solving, okay? Because you all know the correct answer, just that you could not reach the answer by looking at options, you got confused. So search whichever you at least know for sure. Beta-catenin is not required for specification of micromer. Manishri, all this time what did we study then? You are saying micromeres are autonomously specified, which is correct. But then again you are saying beta-catenin is not required. How else then micromeres are specified? Beta-catenin is only the factor, no? Okay. So they are autonomously specified by beta-catenin. You might get confused by WNT signaling, but it is because of the DSH which is present in the micromeres already. That's why the beta-catenin is free. Remember? Yes, DSH is generally activated by WNT signaling. But the DSH here is already present. Remember we studied it is already present in vegetal cortex. I hope you got it now. So beta-catenin is very much required for specification of micromer. D is wrong. So D is wrong means either these two is correct. Anyways, A is correct. That cell fates are determined both by autonomous and conditional. Cell fates means all the cells which are present in the sea urchin. Okay. Now, we know that beta-catenin is required for specification of micromer. It is autonomous only, Vanishri. The specification is autonomous. It is not conditionally specified. Okay. And the factor which is required for that autonomous specification, that is beta-catenin. So B is correct. So B is correct means this is the answer. Rest cells, they are specified by conditional also. Because I mentioned, no, you have the the small micromer, then you have the large micromer. So large micromeres give signal to the cells lying above them to form wedge 2 cells. Wedge 2 cells give signal to the cell lying above them to form wedge 1 cell. That's why, when you take micromeres and you transplant into the animal pole, so there also the micromeres will tell the cells lying next to them to form wedge 2 layer. So from here also gastrulation will start. From here also gastrulation starts. Because micromeres, they themselves get autonomously specified, and then they will tell the other cells conditionally they will specify them to form the other types. Now, how do large micromeres tell that? They produce paracrine and juxtacrine factor. Paracrine means which you release. Cells will release some secretory molecules which will reach the nearby cell's receptor, and the response we will get in the cell. And juxtacrine means a contact is required, like delta and notch signaling. Okay, so just now I mentioned the micromeres, they have a delta signal, and whichever cells have this notch receptor, so this delta and notch signaling will be there, because of which skeletogenic and non-skeletogenic differences in the cells will be created. So with both of this, they will specify the fate of neighbors. No, that is wrong. Only it will be producing the juxtacrine factors. So the correct answer is A and C. An early set of blastomere, B is wrong, C is correct. Paracrine and juxtacrine factors. So an early set of blastomere specification occurs during cleavage in sea urchin embryo. So the result of both conditional and autonomous is there. Only those cells whose fates are autonomous are skeletogenic micromere. Rest all cells are conditionally specified. Micromere produce both paracrine and juxtacrine signal for them to become endoderm. And beta-catenin is essential for patterning the maternally specified animal-vegetal axis in sea urchin embryo. Since D is saying it's not, it's incorrect. Did you get everyone? I hope if any further question comes from this sea urchin topic, you will be able to solve them without any problem. Did you get confidence about part C questions from this topic now? Okay, very good. Okay, one more question is left from this portion. I wanted to discuss that today, but it will take time for explanation. I will discuss that in my next class, next week. That's all from my end, everyone. Thank you for joining in.